My name is Michelle Bravo. Hemoglobinophaties in the fetal position.
Number 1. Gene therapy for hemoglobinopathies.
Living with a condition with no known cure is the devastating reality for those patients suffering from these genetic disorders. Hemoglobin disorders, or hemoglobinopathies, arise when an individual's beta globin gene is dysfunctional. The most common hemoglobinopathies include sickle cell disease and thalassemia, combined affecting an estimated 330,000 kids born worldwide every year. Aside from a blood and bone marrow transplant, there's no cure for sickle cell disease and thalassemia, only treatment for symptom management. But the latest research has brought an experimental gene therapy for a cure in these patients. Adding functional copies of a modified form of the beta globin gene into a patient's own hematopoietic stem cells, the gene therapy gives patients the potential to make functional hemoglobin molecules, thus functional red blood cells. In several ongoing studies, patients with 6 or more months of follow-up after treatment for sickle cell disease had median sickle cell hemoglobin levels reduced to 50% or less of total hemoglobin in the absence of blood transfusions. In thalassemia, studies of the therapy found sufficient hemoglobin production to reduce or eliminate the need for transfusion support among patients who would otherwise require chronic blood transfusions. These results earned the therapy breakthrough designation from the FDA as the first ever gene therapy for either of the conditions. Medical researchers are nearing approval to cure these chronic blood disorders.
Definition. Congenital diseases that result from a mutation or genes on the globin genes on the dot that alter the structure or speed in the synthesis give the globin shine.
In our last video, we discussed the types of blood cells found in peripheral blood, as well as talked about hemolytic anemia, and discussed the various ways in which it may come about. But let's start off by talking about the workhorse of the RBC, hemoglobin, abbreviated HB, in particular its structure. As we learned in the last video, the job of RBCs is to carry oxygen around the body. So let's go ahead and draw our model of an RBC. We'll start with red, and we'll go ahead and draw in a slightly lighter colored center because we know that these are biconcave shape, which means they're lighter in the center when you look under the microscope. And we know that these red blood cells are packed full of hemoglobin, abbreviated HB. So I'll just draw in a bunch of HBs to represent the huge amount of hemoglobin that's in each red blood cell. Now, each of these hemoglobins is actually what binds and transports the oxygen from its loading in the lungs until it can be offloaded somewhere else in the periphery in the tissue. Now, hemoglobin is a tetramer, and we'll model it like a group containing iron. So we'll draw in the symbol for iron, Fe, into each subunit. And the iron is actually what binds the oxygen in the lungs and carries it around to the body. And each one of these can bind an oxygen. So I'll go ahead and draw an oxygen in each one of these subunits. And it's important to note that the hemoglobin is inside the cell, that it should never be outside of the cell, because the reason they're contained is because these iron atoms can cause oxidative damage if they weren't contained inside the cell membrane of the RBC. Within the human genome, there are different genes that code for different subunits: alpha, beta, gamma, and delta. They can all be combined in different ways to form different types of hemoglobin, depending on a number of different factors. As a result, there are many types of hemoglobin, but each of them carries oxygen around the body. Some of these hemoglobin types are better than others at doing their job, simply because the beta subunit, on the other hand, is encoded by one gene only, on chromosome 11. The genes for the gamma and delta subunits are also located on chromosome 11. Let's start off with the three normal variants of hemoglobin, shown here. The first one is hemoglobin A, and the A here stands for adult. Hemoglobin A is a tetramer of 2 alpha and 2 beta subunits, abbreviated alpha 2, beta 2. Hemoglobin A dominates in adult blood, though there is some of a second form of hemoglobin, called hemoglobin A2, floating around as well. Hemoglobin A2 is 2 alpha subunits, but this time they're bound to 2 delta subunits. We'll come back to hemoglobin A2 a little later on when we talk about some of the hemoglobinopathies. The third type of hemoglobin is made primarily in the womb, and it's called hemoglobin F, where the F stands for fetal. Hemoglobin F is made of 2 alpha subunits and
application in nursing. The assessment of nursing by the approach of the 14 reads applied in the pregnancy patient why hemoglobinophaties allow in the know what the need in affecting to define thus dependence of the patient. In order applying nurse care or the patient on the career in the respective treatment is very well administered. Pros and cons. Pros. There is possible of having an effect on show for this in the performance on blood tests for to identify blows in the risk before pregnancy. Cons. Hemoglobinophaties are a hereditary disease to the blood in the transport the oxygen and the cause various disorders including the death of the woman in their baby extreme case.
Hello everyone. So today I'm going to explain you electrophoresis pattern for different hemoglobinopathies. So I have written here normal electrophoresis pattern for hemoglobin, and then different disorders that I have mentioned. I have sickle cell trait here, sickle cell anemia, hemoglobin CAC trait, hemoglobin SC disease, beta thalassemia minor and beta thalassemia major. So let me explain you on like normal hemoglobin electrophoresis. So when you do the electrophoresis, cathode, which is a negative charge, a diode, and anode here, which has got a positive charge. Point of application is here. So depending on the charge, overall net charge of a molecule, so it is going to move. So in normal hemoglobin, so the adult hemoglobin is going to move all the way towards anode. HbF is before adult hemoglobin, and then HbA2, that is minor adult hemoglobin, is almost little closer to the cathode. Now let's see what you are going to see in sickle cell trait. Note that in sickle cell trait, one beta globin is normal, and that beta globin is going all the way towards anode, whereas the other beta globin is of sickle type, that is it has beta or the beta globin, one of the beta globin, sixth position, glutamate is replaced by valine. It means glutamate which has a negative charge, you don't have it now, that's a HbS type. That is why it is moving to the middle and stopping there. Now there is not that much change in fetal hemoglobin and HbA2 in sickle cell trait. Okay, so adult hemoglobin is nearer to the anode, and HbS, one of the beta globin which is changed, it is there in the middle here. Now sickle cell anemia. In sickle cell anemia, both the beta globins, glutamate is replaced by valine at 6th position. Because of that, as you can see, there is no normal adult hemoglobin here, but HbS is there in the middle. And because no useful hemoglobin is synthesized, no adult hemoglobin here, normal adult hemoglobin, so the fetal hemoglobin is increased. That is a compensation mechanism that our body will respond and increase fetal hemoglobin. In the same way, adult hemoglobin HbA2.
Issue applied in the Ecuador. This pathology by the minister of public health news environments. In addition, because they constitute a public health problem, must have in diagnostic prevention in the education programs in the community.
It's not enough to watch the anemia lecture only to know everything about the anemia and the red blood cells defect. You must watch this lecture of the hemoglobinopathies to know everything about the other structural variants of the hemoglobin. So in our today's lecture of the hemoglobinopathies, we are going to cover the different structural variants of the hemoglobin other than the thalassemia. So let's see that what we will talk about in our today's lecture. We will first go through the question, what is the hemoglobinopathies? We will discuss the general signs and symptoms of the hemoglobinopathy. Then we will talk about the different types of the hemoglobinopathies, the different structural variants of the hemoglobinopathy that are important and that we will cover in our today's lecture. Moving on, we will start with the sickle cell disease. We will talk about the progression of the disease, how it defect in single gene, the homozygous defect will lead to the thrombosis, the production of the sickle cell as well, and we will discuss that how the production of this thrombosis will then cause the aplastic crisis, and also it will cause the sequestration crisis, how it can lead to the different organ failures, and what are the clinical features that it will cause. Then we will move on toward the pathology of the sickle cell disease, that how you can differentiate the sickle cell disease from other red blood cells defects. Then we will talk about the sickle cell trait, that what is the difference between the sickle cell trait and the sickle cell disease. Then we will also talk about the double heterozygosity for the hemoglobin S. We will talk about the difference between the homozygous, heterozygous, and also the double heterozygous conditions of the sickle cell disease. Which one is more severe, what are the symptoms that will occur, and what is the severity of all of these three types of the diseases. Then we will talk.
Conclusion. The frequency in the variant found in the study confirmed the hemoglobinopathy represents a public health problem on the importance of systematic screening program in the hemoglobinopathy that include in the early treatment, but also genetic education on counseling program in the for family group.
So here's a picture of mom and a little fetus. And at this point, when the fetus is still attached by the umbilical cord, everything that goes into the fetus is really originating from mom. She controls all the nutrients and all the oxygen that goes into that baby. And with oxygen in mind, there are a couple of interesting ways that the baby, in this case, this little fetus on the right, has come up with to be able to get as much oxygen as possible from mom. Because remember, the fetus is trying to grow, and it wants to make sure all of its tissues that are growing and developing have enough oxygen. So there are a couple of tricks. And the first trick, let me actually just draw it out for you, is simply looking at a single vial of blood. If we look at a single vial of blood from mom and compare it to a vial of blood from babies, let me try to draw the vials about the same height and width. These are the two vials. If I was to take now, let's say, a little bit of mom's blood and spin it down, let's say in this little tube, and then do the exact same thing with the baby's blood, take some of baby's blood and spin it down, that spun blood, once it's spun down, would actually separate out into little parts, right? You'd have three different little layers, and this first layer would be something like this. This is called the plasma. The next layer right below it, remember there's a little layer of white blood cells and platelets.
And below that, right here, is a layer of red blood cells. And remember, red blood cells are the ones that contain the hemoglobin. They're the ones that are going to move oxygen around. And in mom, the percent this red layer takes up is about 35%, meaning this whole thing would be 100%. Let's say this entire thing would be 100%. And of that, just over 1/3, or 35% exactly, is that bottom red layer. That's the red blood cell layer. So we would call this the hematocrit, right? So this is mom's hematocrit, and this is a very typical number for a pregnant woman. It varies depending on, whether you're a man or a woman and what age you are. But for a pregnant woman, 35% is a pretty reasonable number. Now going over here to the baby side, let's draw in what baby's blood probably looks like. The baby has a lot less of the blood taken up by plasma, so that layer is going to be smaller. And then the next layer, the white blood cell layer, that's a very small layer anyway, so that's not going to change much. And the final layer, the third layer, is a red blood cell layer. This layer takes up about, let's say, about 55%. So I hope I didn't kind of misdraw that, but that's about right, about 55%. So here, the hematocrit is much higher. Now, what does that mean? If the hematocrit is higher in the baby, about 55%, then that means that the baby actually has more red blood cells going around in a given amount of volume of blood, and those red blood cells then can take up more oxygen, because that's really the part of blood that we care about when it comes to moving oxygen around. So that's one trick. In terms of tricks for getting more oxygen, simply having more of the red blood cells in a given volume of blood is kind of the amount of red blood cells is going to go up in the fetus. And that's kind of one trick. When I say trick,